Hi everyone, it's a pleasure to be here with you today to present to you the work of my colleagues and I, what we've been doing for the past few months. As you can infer from the title of the presentation, I'm not in bioinformatics per se. I'm a computer scientist, but I apply it to medical imaging. We mostly use magnetic resonance imaging, or MRI. If you've never really heard about MRI, well, read the news. It's a bit everywhere right now. It's kind of a big modality in medical imaging, but don't worry, you don't need to know anything about it to be able to follow the talk. I'm working at the Sherbrooke Connectivity Imaging Laboratory, or SCIL for short. We're a small but productive team based in Sherbrooke, Quebec, Canada. We're one of many labs around the world who are trying to explain how the billions, hundreds of billions of neurons inside the central nervous system connect together to create what we know as the human experience. We are part of an international community analyzing diffusion MRI with state-of-the-art processing, image processing, and statistics to reveal the structure and geometry of axonal bodies and their projection throughout all of the central nervous system. Our combined goal as a community is to reconstruct a connectome, a map of those neuronal connections, using the structural information that we get from diffusion MRI, and to also use all the knowledge that we gather across the years to diagnose top pathologies that are still affecting humans throughout their lives. It's an intensive task. Multiple key steps must be done correctly for diffusion MRI images to be analyzed afterwards, and most of them require long computing times. MRI images are spatially dense. We're talking about millions, sometimes tens of millions of data points. And in diffusion MRI, we don't just do one of those acquisitions. We repeat that tens or hundreds of times to get enough data to fit our models. And thanks to compression, those images fit on disk nicely, but we have to be clever when we process those because they tend to break the available RAM on our computers. And it's not all. Our goal is to reconstruct neuronal pathways, and so we use an algorithm called tractography, which produces millions and millions of potential connections, each sample at tens to hundreds of 3D points. And those we further analyze using filtering algorithm, clustering. We fuse those with other type of information, most of the time in resource-hungry, really high-dimensional frameworks. So I don't think I need to say more on that. Diffusion MRI is really hard on the computer. It tends to break in most HPC or even local setups that we have. But when we're clever, we do the work well. And so to do that, the whole community around diffusion MRI has created efficient tools, robust and well parallelized, as well as good quality control solutions, and other solutions to benchmark our algorithms against each other. It's a communal effort, really stemming all around the world. Pretty much every lab has also developed GUIs to help its users, develop pipelines to make it more efficient. The other one. Here in the lab, we've adopted Nextflow quite early in the DSL1 era, beginning of DSL2. It's what I like to call our monolithic era. We put up a lot of good pipelines. I think we're at 19 right now in the lab. It's a crazy huge number. But they work good. We have good notoriety with those pipelines. We use them locally, but also share them with collaborators in experimental neurosurgery or for pharmaceutical applications. And we had great success. But there is always a problem. In fact, multiple problems with monolithic codes. It's hard to the maintenance is hard. They are hard to manage. Validation is a complete hell. If you don't do the things right, you end up just repeating yourself over and over the parts of code that are just replicating everywhere. And in case of Nextflow pipeline, it's not really efficient as well, because we're batching multiple pipelines one after the others, and we're not using the Nextflow engine. We're usually using Bash, which is not really good. And all of those problems have been creeping on us throughout the years, but luckily, we found nf-core just in time to solve all of this. And so we put together a small team, 5 people in our lab, probably the craziest people in the world, but yeah, most assuredly the best fitted for the task of transitioning our whole pipeline infrastructure into nf-core. They had 7 months up to today, and I'm pretty happy to say that the task was successful. I'm here today and I'm able to present to you nf-scil. The initial goal that we had was to take all the common parts of our pipeline and put it inside of the nf-core framework. And so for that, we created a big compendium of modules. It was quite easy, given that nf-core has a really good documentation and a really good tutorial that you can follow. We also created a whole collection of sub-workflows using those modules. And we validated everything thoroughly and generalized it. In fact, it's way better than what we had before. Our whole collection of pipeline was not really tested at all. But now we have that through nf-core. Yeah, we hit a few hurdles along the way. Recently, we did the switch from PyTest to nf-test, but I think that we're not alone on that. And even then, the road has been quite smooth. So smooth, in fact, that in addition to nf-scil, we already ported our biggest pipeline in the lab, TractoFlow, to the nf-core framework. And while initially TractoFlow took a lot of time to develop into its fully-fledged form, this transition took less than a week to do. And that's really hopeful for us if we want to streamline the adoption of Nextflow in our field. But however, while we were at it, we realized that our job could be more and should be more than just those additions to nf-core. Like I said at the beginning, we're kind of remote from bioinformatics, and we think that our contribution should also come with the expertise we grew throughout the years, locally in our lab, but also throughout our international network of collaborators. And so what we want to do is to rebrand our project into nf-neuro, not to form a community aside of nf-core, but to create a community connected to it, a little more close to our field of neuroimaging. Because right now, in terms of adoption of Nextflow in neuroimaging, it's really low. I did a little search on PubMed before coming here. I searched for neuro and Nextflow while removing genetics, bioinformatics, and things like that. And right now, there's 2 papers. The first one, TractoFlow, has been developed in our lab, and the second one, VersaFlow, is the flow that I developed in my master's studies. And if you remove the last few keywords after genetics, you get a little more pipeline, but they're not applying in medical imaging. And so the plan for that is to start locally. We want to create an expertise in our network and use it to lay core foundation for neuroimaging using Nextflow and nf-core. Yes, we want to provide robust modules and sub-workflows, but we also want to provide them with good computing configuration for clusters and the cloud, as well as starting to produce educational content adapted to our community. The expertise that we have in the lab is mostly in diffusion MRI, but we've touched to CT and PET imaging as well. We have some collaborators that use functional MRI. And while each of those neuroimaging fields have their specificities, there is also a lot of commonalities between all of the processing that they do. And I think that those commonalities for us is a target well inside our range. We believe that using nf-core and Nextflow in neuroimaging will make the field even better, more robust, more efficient. but also will increase the corpus of knowledge that we have accessible for education. We also believe that it profits both ways, and that our addition to the NF core community will make it better. We have different kinds of problems, different kinds of requirements, and I think that we can definitely help make NF core even better. And on that, I want to thank you. I want to thank my team that you probably have seen a little going around. They all have these beautiful shirts. I want to thank my lab, the SIL, as well as our funding. Thank you very much.
Thank you, Alex. It was a wonderful story, a wonderful success story, I think. And also compliments on your shirts. That's fantastic. I feel like we should have that on the community team as well.
Yeah, they're stuck with
just imitation is the most sincere form of flattery, just so you know. That was fantastic. Can you clarify? You might have said it. I might not have caught it. But you want to nucleate this community within your field. Are you thinking of doing that through GitHub organization that's separate from NF core and have most of the code there, but have some imports from NF core, or fold everything under NF core? I'm curious, and I'll preface that by saying we definitely don't want to make it sound like you have to put everything in NF core. Yeah. I think there's a future world where there's more a connected, like federated ecosystem of these kinds of communities, and I kind of am interested in your vision for how these things connect.
Yeah, thank you very much for the question. In fact, in the past 2 days in the hackathon, we had very, very nice talk with the NF core team. And while we'll still have this kind of NF neuro locally to play in, break, while not breaking the whole NF core framework, we've been talking with people, and we're starting right now a special interest group for neuroimaging. We really want to get the community around and in NF core as soon as possible, because we think it can really help accelerate the whole research field so much.
That's excellent. Thank you very much. Thank you, Alex.